Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated.
Every single living thing, from you and me to the smallest bacteria, has this incredible blueprint inside its cells, right? It's the code of life, the DNA. But actually getting your hands on that code, well, that's like trying to pull one super delicate thread from a huge, tangled, messy ball of yarn. So how in the world have scientists been doing this for decades? Let's get into the classic and, frankly, still super powerful method they've been using all this time.
So yeah, that's the big question, isn't it? You've got the DNA, but it's wrapped up in membranes. It's all tangled up with proteins, and it's swimming around with all sorts of other molecules like RNA. How do you possibly get it out, and get it out clean?
Well, it turns out the answer is some seriously clever chemistry. We're talking about the Phenol Chloroform extraction method. It's a legendary technique that uses this special chemical cocktail to, well, elegantly separate the good stuff, our precious DNA, from all that cellular gunk.
All right, so our first mission, should we choose to accept it, is to break in. And this isn't some gentle knock on the door. No, this is a full-on molecular heist. We've got to crack open the cell and get past all its defenses. In science, we call this cell lysis.
And you know, any good heist needs a specialized toolkit. So first up, we've got SDS. It's basically a detergent, and its job is to dissolve the cell's fatty membranes. Just think of how soap cuts right through grease. Next, we send in the cleanup crew. We've got Proteinase K, which is like a little molecular Pac-Man that just gobbles up all the proteins. At the same time, RNase goes to work shredding any RNA we don't want. And finally, we need security, right? That's EDTA. It disarms these nasty enzymes that would try to destroy our DNA, making sure our prize stays perfectly intact.
If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated.
Okay, so once the cells burst open, everything is just floating around in this chaotic soup. This is where the real magic happens. It's time for the great separation. And it all hinges on a really simple, fundamental idea in chemistry.
And that idea is called differential solubility. You know how oil and vinegar in salad dressing always separate? They just refuse to mix? Well, it's the exact same principle here. Molecules are kind of forced to pick a side. Are you going to hang out in the watery aqueous layer or the oily organic layer? We use this exact preference to sort through everything in our cellular soup.
So when we add our phenol and chloroform and spin the whole thing in a centrifuge, this is what we see. 3 totally distinct layers. Down at the bottom, in the oily organic phase, you get all the fats and proteins. Then, floating at the very top, in the watery aqueous layer, that's our prize. That's the DNA. And stuck right in the middle, you get this gross, white, gunky layer of junk called the interphase. The whole reason this works is that DNA's chemical structure makes it absolutely love water, so it stays safe and sound up top.
All right, so we get the theory, but what does this actually look like when you're at the lab bench? Let's just do a quick run-through of the protocol in action.
It really boils down to 5 key moves. 1. Lysol. We crack open the cells, just like we talked about. 2. Separate. We add our organic chemicals and spin it all down to get those nice layers. 3. Collect. Now this is the tricky part. You have to use a pipette to carefully suck up that top watery layer without grabbing any of the gunk from the middle. 4. Precipitate. This is the coolest part. You add ice-cold ethanol and a little salt, and all of a sudden the DNA clumps together and becomes this visible, stringy, solid mass. It's awesome. And finally, 5. Purify. We give it a quick wash, and we're good to go.
Now, this method is obviously super powerful, but it's not all sunshine and rainbows. It definitely comes with a price, and that price is some pretty serious hazards and a few common headaches for anyone who does it.
And this right here is the fundamental trade-off. On one hand, you get a ton of really fantastic high-quality DNA. It's super cheap to do, and you can easily scale it up for big samples. But, on the other hand, you're dealing with some really nasty chemicals, it takes a long time, it's a lot of hands-on work, and you always run the risk of getting a tiny bit of phenol in your final sample, which can just completely ruin your future experiments.
If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated.
And look, I cannot stress this enough. Safety is 100% non-negotiable here. Phenol gives you horrific chemical burns. Chloroform is toxic. You absolutely must do this procedure inside a chemical fume hood, no exceptions. And you need to be wearing the right gear. Gloves, lab coat, splash goggles. Seriously, you don't mess around with this stuff.
And you know, even when you're being super safe, things can still go wrong. The number one problem people run into is just not getting enough DNA out at the end. You can also end up with leftover protein or RNA if those cleanup steps weren't perfect. And if you're a little too rough with your mixing, you can actually shred the long DNA strands into little pieces. The good news is, for every one of these problems, there's a well-known fix.
So, you might be wondering, with all these hazards and all the fancy new kits you can buy, why on earth are we still talking about this old school method? Well, because despite its flaws, this classic has a pretty amazing and lasting legacy. Let's talk about why it's still so important.
The biggest reason it's still around is just quality, pure and simple. And scientists have a way to prove it. They use a machine called a spectrophotometer to check two key numbers. An A260/280 ratio of about 1.8, boom, that tells you your sample is incredibly clean with almost no protein contamination. An A260/230 ratio above 2.0, that means you washed away all the nasty chemical gunk. And this method, when you do it right, absolutely nails these numbers every time.
And that level of purity is why this DNA is considered the gold standard for some of the most cutting-edge science out there. I'm talking about things like sequencing an entire genome, building massive genetic libraries, forensic analysis, even pulling ancient DNA out of dirt to study whole ecosystems from the past. These kinds of projects need perfect, long strands of DNA, and they need a lot of it, which is exactly what this method delivers.
Download our Microbiology study app made for researchers like you.
And that really gets to the other big reason it's stuck around for so long. It's cheap. For labs that are on a tight budget, or for scientists in places where it's hard to get those expensive commercial kits, the fact that you can do this with basic chemicals is an absolute game changer. This quote here really just sums it all up perfectly. It holds its ground because it gives you top-tier quality for rock-bottom costs.
So, all of this leaves us with a pretty interesting question. Is the phenol chloroform method a revered laboratory classic, the absolute bedrock of molecular biology, or is it kind of a toxic relic that we should have left in the past? The truth, as usual, is probably somewhere in the middle. It's an incredibly powerful tool, but one with a dangerous edge, and it demands respect, a lot of skill, and a very, very good fume hood.
Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you.